Welcome to Beyond Lab Walls, a podcast from the Salk Institute. Join hosts Isabella Davis and Nicole Milnarek on a journey behind the scenes of the renowned Research Institute in San Diego, California. We're taking you inside the lab to hear the latest discoveries in cutting-edge neuroscience, plant biology, cancer, aging, and more. Explore the fascinating world of science while listening to the stories of the brilliant minds behind it. Here at Salk, we're unlocking the secrets of life itself and sharing them beyond lab walls.
Welcome back, listeners. I'm Isabella, and today I'm joined by Aksinya Derevyanko. Aksinya is a molecular biologist currently working in the lab of Professor Nicola Allen here at Salk. She's interested in the microscopic connections between the neurons in our brains called synapses. These synapses are where messages get exchanged between neurons and are the foundation for healthy brain function. Now, Aksinya is asking what happens when these synapses dysfunction, and in particular, does that dysfunction relate to Alzheimer's disease? And if so, how? But before we get into all of that and her science, let's go back to the beginning of her story. I'm very excited to be chatting with her today. Welcome to the podcast. Tell me, where did you grow up?
So I was born still in USSR, and I grew up in Altai region. And so in my family, my parents were not really researchers. Well, my dad, he's like a creator. He's an artist. He's a chemical engineer. He also did like a line to recycle and repurpose plastic. So he's just like all over the place. And my mom, she was a mathematician. But when USSR collapsed, many institutions, they had to downsize, especially in Far East. So many scientists had to repurpose. So my mom did. And I kind of benefited from that because most of my teachers in high school were like nuclear physicists or plasma physicists. My parents put the importance of education because education in my country is very accessible to everyone and it's almost free. So you just need to study. So their approaches were very interesting, I think. First, they got me enrolled in a job, so like service-oriented. So at like age of maybe 13, I was working 24-hour shifts at the gas station, which I was totally on board with. And I think it was a great experience, which also helped me understand how hard is the job market, that some jobs are lower paid than others. And if I want to do some more creative type of things, with all my respect to gas station workers, I have to pursue my education. And another fun thing they did is that they went and did a master's degree to show us that you should always study personal development, and it was fun to go to school the same time as them.
That's so funny and sweet to be in school with your parents. I would have loved that. So you knew you wanted to focus on your education, but did you know you wanted to do science?
I wanted to become a linguist. I didn't think of being a scientist just yet. So I tried to enroll in Moscow State, and I went there and did my exams like 3 times. And with the system where you can study for free, you need to compete sometimes with the whole country. So I couldn't get in, which I fulfilled that linguistic dream later on since I travel and learn a couple of languages. So then my plan B was, I didn't really have a plan B. So I was going by method by contradiction, let's say. So take into consideration, we finished high school pretty early, so I was like 16. So it's very early age to take that important decision. So I was thinking what I don't want to do and don't want to pursue. And then life sciences sounds interesting because it's about life. It must be exciting. So I considered Novosibirsk State University, which is famous in Russia for its departments in STEM. I passed entry exam, I got admitted, and then I went to admission committee. And it is important to have a really good friend because I brought my friend with me. And when I come in, they say, congratulations. So what department do you want to join? Biology or chemistry? I look at my friend, a very good friend, and she said, of course, biology. So I turn around and say, of course, biology. And here I am.
That's so funny. The best way to make any decision, just relying on your friends. So you started in biology. And then how'd you end up interested in neuroscience?
Right. So as soon as I got into university, I quickly realized that all my peers are very familiar with what is to be a researcher. For example, many of their parents were researchers in the institutes around, or there was high school associated at that university. So as soon as I could, I tried to get my first lab experience. So the first lab I actually got in after the course of botanics was in a Siberian botanic garden. I did a project there and in the Institute of Organic Chemistry where we study mushrooms and we prepared different types of extract. My studies were going on and I realized I'm really excited about molecular biology. So I talked to my PI at the time, explained her my interest, and then she introduced me to her friend who was running a lab in the institute across the street, which was Institute for Chemical Biology and Fundamental Medicine. And my first molecular biology PI, Zharkov Dmitry Olegovich, he introduced me to the biology lab world. He taught me all the great skills, how to design the experiment, the lab ethics, how to approach your time management and all sorts of things. So I'm very grateful to him for that. And then he was also the one that introduced me to the synapse, which I'm actively working on right now, which is the synapse is this connection where neurons transmit signal one to another. So he explained what it is for us in the course of molecular pharmacology as an example with Botox. So I was working on the project where I look how DNA repair mechanism can lead to expansion of 3-nucleotide repeats, which is a trigger of many neurodegenerative disorders such as Huntington disease. And so we didn't have a class of the neuroscience at the university, but that sparked a lot my interest. And since then, I always wanted to know more about how the brain works and all these diseases develop. And then my first PI, he also presented me with amazing opportunity, and I'm so thankful for that. We had a friends lab in Villejuif near Paris in France, which were also working on the DNA repair. So I could do 3 internships and do portion of my experiments in their lab. So I was able to visit International Institute and see how differently things work. And I quickly realized that for me, it is very important to have these diverse experiences in science and in life in general, which was not necessarily how things were working back in Russia. But I realized it's quite important to learn as many things as you can and to see all different labs, meet different people. After having this realization that I want this international expertise and that I'm interested in translational research, I started to apply for PhD fellowships abroad. And I got one actually in Spain, in Madrid. So that's where I first moved to do my PhD before coming to the United States. And the lab in Madrid, it was a great lab that was working on telomere biology in a context of aging, cancer, and other diseases run by Dr. Maria Blasco.
And what are telomeres?
So telomeres are these repetitive sequences that form caps on the end of the chromosome. Because if you think the chromosome is like double-strand DNA, so if they are not hidden, they can be recognized by DNA repair machineries, and this can trigger DNA repair. So chromosomes can be fused, or it can signal that there is a damage and cell will go to senescence and die. So it's very important topic in the field of aging.
So then you started doing this telomere research in Spain, in Madrid. How'd you go from Spain to San Diego?
Right. So not in my main project, which I did on aging, I also did several collaboration within the lab and other institutions as well, where we touch on neurodegeneration. And this reminded me about my spark in neuroscience research. And then I started to look for a lab in neuroscience, which are the people who are looking at all these different cell types in the brain and in the context of health and neurodegenerative diseases. That's how I got here at Salk and got into Nicola Allen's lab. Actually, I learned about San Diego during my PhD. At first, I met a friend who was doing her PhD at Scripps and later her postdoc at UCSD. We met at Cold Spring Harbor conference in China, so she told me all great things about San Diego. And then a person came to my lab who was a lab manager here at Salk, and then she told me all great things about Salk, so I didn't hesitate and applied right away and got so lucky that Nicola was interested.
That's so cool. You went from kind of general biology to the brain to neurodegeneration and DNA mechanisms and telomeres and synapses, more and more specific as you went on until getting to Salk. What exactly are you studying now?
We are interested in these cells called astrocytes, which are star-shaped glia cell, which are very important for central nervous system, and they play very important functions such as providing trophic support to neurons. They regulate recycling of neurotransmitter, the molecule that neurons use to communicate to each other. They also form a part of blood-brain barrier, this barrier that filters what ions and molecules can go from the blood inside the brain. And also, astrocytes, they release different molecules that can trigger synapse formation, and then they wrap around the synapse and regulate their stability and strength. Astrocyte, by releasing different molecules, can actually trigger the increase of a very different receptor at the synapse, so the regulation is very specific. And I'm working on one type of these molecules. They secrete this molecule, increase of a certain receptor, which characterize a stable synapses. And this molecule is actually regulate establishment of synaptic stability in development. Interestingly enough, in Alzheimer's disease, this type of synapses diminish. So we thought, great, we can get this knowledge from the development and apply to Alzheimer's disease context. So what I do in my project, I overexpress these molecules in the astrocytes in the models of Alzheimer's disease. Astrocytes secreted in the synapse increase number of that receptors and make synapses stronger. And I see if I can prevent or rescue synapse loss and if this can have effect on memory and cognition as well.
So are you looking at models of Alzheimer's disease, or are you just looking at aging models?
I'm working specifically with models on Alzheimer that also represent different hallmarks, such as tau tangles or amyloid plaques.
Tau tangles and amyloid plaques are two different protein blockages that are characteristic of Alzheimer's disease. Tau protein tangles tend to occur inside neurons, while amyloid protein plaques clump up between neurons. So in addition to looking at these hallmarks of Alzheimer's, you're also finding out a lot of things about synaptic strength and synaptic loss. Are those things that can be applied outside of Alzheimer's to help understand the brain more generally or how memory works?
This is definitely very important in translational research because not only Alzheimer's but other neurodegenerative disease, they also experience synapse loss. Mm-hmm. So it can be applied to many conditions. The molecule that I'm using will be applied specifically where these types of receptors are lost. And yeah, of course, doing this research, I learn all about memory, how it forms, how it stabilizes, and how it's lost in disease.
What, to a neuroscientist, is memory?
So all of us, we have new experiences, we visit new places and meet new people, right? So the memory is the ability to retain that information over time. And memory can be studied at different levels: psychology, anatomy, electrophysiology, and molecular biology. So me, as a molecular biologist, I'm looking at the synapse, which is a key player in memory formation. So when learning and memory is associated with activation in the brain upon experience. So neurons become activated, and then using neurotransmitter, they transmit signal one to another. A neurotransmitter is released, is bind to receptors on the next neuron. They open, allow the ions in, and cause all these molecular changes, which in turn can change the structure of the synapse and make it more stable and responsive. Upon memory recall, in response to a specific cue, such as smell, an image, or a sound, this network that was reinforced will be activated. And when synapses are weakened or lost, it's where the memory is affected. And this can happen naturally, like when we are forgetting or in disease, when we don't want to lose this memory. Unfortunately, all these pathological processes, they interrupt this healthy synaptic function.
So when you're looking at this kind of stuff at Salk, is it helpful to have all these different disciplines working together, immunology, neuroscience, all these different backgrounds, even just in Nicola's lab, not even considering other labs that you could be talking with members of that are your friends during lunch? Do you find that helpful when you're approaching questions and doing the research?
Yeah, no, this is great because in our lab, everybody has such a different expertise. We have immunologists, electrophysiologists. I'm a molecular biologist who came without any knowledge to neuroscience. So it's very impactful to discuss all these things with each other, and on the lab meetings, somebody has more expertise with experiments on the memory, someone with the genetics. So it's very helpful, and we all collaborate together on each other's project, try to get the best knowledge as we can.
Yeah, it's one of my favorite things about Salk because everyone seems to work together. It's very cool. And is there anything you're working on right now that you're really excited about?
Currently, I'm analyzing all this data on the effects that we have on the synapses. I'm also looking at the neurons and other glia cells, and as well as analyzing data from behavioral experiments where we seem to be seeing some encouraging effects. So I'm very excited about that. And at my last years of the postdoc, I'm also trying to expand my tools and the models that I'm working in on looking at Alzheimer's, not only in the models that I'm working right now, but also with human models. I think it's very powerful. And research have developed a lot over past years. I'm not working on that right now, but one of my biggest interests is to look in the Alzheimer's in 3D human organoids cultures, which resemble area of the human brains, right? And this is powerful, not just because it's just human cell, but also thinking that usually we use the transgenic models, which carry the familial mutation. However, in humans, familial mutation account maybe only for 5% of Alzheimer's, whereas the rest, 95, are sporadic cases. So taking human cells from a patient with sporadic cases, we can develop organoids and mimic some of that, model some of that pathology. I got lucky and got selected, and last year attended the Stanford Human Brain Organogenesis workshop. It was great hands-on workshop where they taught us all the lab work and all the applications. I hope I can do a little bit of that research in here and maybe establish some collaborations.
And I think it's cool to see basic research and medical research kind of moving in the same personalized direction, keeping those epigenetic markers, like you said, on these models. That's a huge step. It's very exciting.
I'm actually also really excited that in our project, we're looking at really these early stages of disease progression, which is very important because earlier you can target the disease, maybe more chances of a successful outcome you can have. And it was a great breakthrough with all these drugs that were approved by FDA that target amyloid. I think since Alzheimer's is such a complex disease, it would be cool to combine those with drugs that target other aspects of the disease progression. For example, accumulation of tau, which pathological tau can actually in many ways impair synaptic function, or combining it with approaches that I'm developing right now to increasing synaptic strength and combat the disease.
Do you think the future of Alzheimer's care is going to be preventative rather than reactive?
Well, since majority of the cases are sporadic, I think this may happen if we understand all the genetics and all the risk and protective factor. Then maybe we can have a better prediction which people are at risk.
So what are your plans next after Salk?
So since I'm at the last year of my postdoc, I'm trying to get the best knowledge I can and develop my skills as much as I can. And I'm really interested in continuing my career as a researcher. I will try to, apply as an PI for an academic position this upcoming years. Honestly, I'm quite open to work in the academic setting or more industrial of biotech, because my interest is really keep working on memories, Alzheimer's, translational research. So I think I can benefit in both. But I guess the first choice will be in academia, because I like how it allows you to be creative as a scientist. And I've never been on the other side, so I don't know.
Do you ever think about teaching or what you would want to teach if you were to be in an academic setting and in a classroom?
Honestly, I have never had the experience teaching at the class, so I don't know if, honestly, I'd love it or not. I don't know if you can tell, I fear public. But actually, I honestly enjoy working with people one-on-one. And this is another great thing about Salk, that we have students that are coming, undergrad students that are coming from UCSD. And I had a chance to work with many undergrads, and I really enjoy teaching them, passing them my knowledge and my expertise. So maybe.
And what kind of stuff do you get up to off campus? What's life like for you in San Diego?
Well, there is a whole another life. Well, I like to do many different things. One of the biggest thing I'm into, I'll call it semi-professional dancer. I started to dance ballet when I was at school. And then at the university, I danced urban dance style, like a hip hop and funky styles and breaking, which I still do. But then during my PhD times, I got involved in a Latin dance, salsa and bachata, and I grew a lot in that world. And at some point, I started to teach class with my former dance couple, which was a PhD student from another lab across. So currently I'm not teaching, but I'm in 2 dance companies. And actually I have 4 shows this weekend, and one is today.
4? That's crazy.
Yes. So I'm currently working on development myself as a dancer, in my free time, of course.
That's great. Are you hoping to keep that up? Are you going to keep dancing?
I think so. I really enjoy it, and I think it helps me actually be a better scientist, because both fields are very creative but also require a lot of mind work. Then it's nice to have a balance between sedentary lifestyle. Then it also helps me in these things like as public performance. And also I think an interesting aspect for me is that, sometimes a hard experiment didn't work or something went east. So I always have that other side that makes me happy. And when sometimes I don't do a good performance, I have my experiments I'm so excited about. So it kind of helped me to feel good level of happiness.
Well, that is a beautiful note to leave things on. Staying happy, feeling good. Thank you so much for being on the podcast.
Thanks for having me. My pleasure.
Aksinya is such a fantastic example of what makes Salk science special, especially when it comes to Alzheimer's. And that's the combination of interdisciplinary collaboration and basic research. Interdisciplinary collaboration creates a give and take and push and pull of information that shapes questions only Salk scientists can ask. And these one-of-a-kind questions mean one-of-a-kind answers, bringing us closer to understanding how our brains age and what disease progression and prevention may look like. Basic research means we aren't just enhancing what we know about a single disease or disorder. Aksinya is going back to the basics, looking at synaptic dysfunction that impacts all of us as we age. The further she digs, the more we know about how the brain works for everyone, how memories come and go, how our brains change over time, and how we might prevent these general functional losses. The success of this collaborative, basic research relies on scientists that are as friendly as they are talented, Speaking with Aksinya was a tremendous reminder of that, and I'm excited to see what she discovers next.
Beyond Lab Walls is a production of the Salk Office of Communications. To hear the latest science stories coming out of Salk, subscribe to our podcast and visit salk.edu to join our new exclusive media channel, Salk Streaming. There, you'll find interviews with our scientists, videos on our recent studies, and public lectures by our world-renowned professors. You can also explore our award-winning magazine, Inside Salk, and join our monthly newsletter to stay up to date on the world within these walls.